Today, we're going to shrink the spotlight and find out why it is so incredibly difficult to swat a fly, even if they're taking a snooze. Welcome back to Sketchbook Biology. When we mammals sleep, our brains broadcast slow, synchronized electrical waves called slow wave activity. And these waves are a hallmark of deep sleep, often associated with the dulling of senses and reduced motor activity. Now we have this fancy egg-shaped thing in our brains called the thalamus to manage generating these waves. But recently, scientists have found out that even fruit flies that definitely do not have thalami, also generate slow wave activity during undisturbed sleep. Now, every living creature needs a bit of deep sleep, even if their life's purpose is to be a global nuisance. But an insect with a brain the size of a grain of salt, showing the same brain activity as a human during sleep, opens a window into a very interesting area of research. Because the possibility of getting unceremoniously eaten or flattened while sleeping is significantly higher for a fly than it is for a human. How does an animal tune out the world enough to sleep and get the full regenerative benefits, but still stay alert enough to respond to external threats? And that is what a team of researchers from the Charité Medical University, the Humboldt University, and the Free University of Berlin, and several other very distinguished institutions set out to investigate. Now we know a lot about fruit flies in general. The fruit fly, Drosophila melanogaster, is an extremely well-studied model organism. Inside the fly brain there are three distinct neural networks. The R5 is a ring of neurons along a major visual sensory pathway. Then we have the dorsal fan-shaped body, and finally a set of cells called helicon. These pathways together coordinate sleep and sensory processing. And to understand how fruit flies slip into slow wave sleep, the scientists used imaging and some genetic and behavioral experiments to record what goes on in these pathways during fruit fly nap time. So the methods described in this paper are fairly dense, and I'm definitely not a neurophysiologist, but I'm gonna try my best to try and explain to you as clearly as I can. So please bear with me because the science here is so incredibly cool. The flies that were used for the study were grown at a comfy 25 degrees Celsius with 60% humidity, and when the study began, most were 3 to 10 days old. The researchers started their investigations by using high-speed imaging to record the faint voltage and calcium changes in specific brain cells along the three pathways that we talked about earlier. Next, they moved on to optogenetic experiments. Maybe it's because I haven't read too much into this type of science, but I found this technique so interesting. Optogenetics refers to the use of light to control the cells of living beings. Crazy! Now this involves first genetically engineering the target cells to produce the light-sensitive proteins. And for this study, the scientists were using a specific genotype of Drosophila, in which certain neurons make light-sensitive proteins called CS crimson and GTACR1. Neurons that express CS crimson are activated by specific red wavelengths, while neurons expressing GTACR1 are inhibited by specific green wavelengths. Sometimes I'm reading these articles and I'm thinking, how did you guys even find these things out? I suppose when you know so much about a model organism already, doing stuff like this must be like just another Tuesday. These optogenetic experiments also required flies to eat food supplemented with all-trans retinol, so those light-sensitive proteins would work as intended. And once they've had that food for a while, the flies were filmed doing just normal fruit fly things in infrared lit stages or in glass tubes. The researchers then compared this baseline activity to activity during red and green light stimulation to measure changes in speed, walking time, resting times, and responsiveness to sudden lights or puffs of air. And to get the most detailed neural information, they carefully inserted very tiny electrodes into single R5 neurons. Yeah, very, very small, to measure electrical activity while stimulating those neurons with red light pulses. And finally, they used all this raw data to create and inform computational network models and other brain mapping techniques to explore how these neurons connect and create slow wave oscillations. Okay, now for the results. The researchers discovered that the DFSB, this is the dorsal fan-shaped body we talked about earlier from those 3 neural pathways, produces strong slow wave activity at night, and it synchronizes with the R5 network. In the optogenetic experiments, activating the DFSB triggered the R5. So activating the R5 network made the slow wave activity from the DFSB even stronger. And silencing the DFSB reduced the slow wave oscillations from the R5. So the R5 and the DFSB are mutually reinforcing each other. Slow wave activity also showed very strong behavioral effects. Activating R5 in flies that were already awake produced a kind of passive state. They sat and groomed quietly but were still able to be startled by strong puffs of air. In addition to all that, short green light flashes woke sleeping flies depending on stimulus duration. Now this is cool because the fact that the stimulus duration has an effect at all tells us that the internal natural sleep drive is fighting these external light cues. And this is where the helicon comes in. Okay, follow me closely here, it's going to be a doozy. When the researchers pulsed the red light to activate either the R5 or the helicon neurons in sleeping flies in separate trials, while also having the green light pulsing, the activation of the helicon quickly woke the flies up. But activating just the R5 did not wake the flies up. When both sets of neurons were turned on together with a slow once per second or 1 Hz light pulsing rhythm, the flies became even more still than usual and ignored both the green flash and a puff of air. Also, inhibiting the helicon cells alone also induced that passive state. This tells us that the R5 can override the get-up signal from the helicon cells, and that when the two fire in sync, they create a neural filter that blocks out most sensory input. The brain mapping work that the researchers did gives us even more context. Both R5 and helicon neurons connect to EPG neurons. Now these EPG neurons normally help a fly sense its direction as it navigates. The R5 and the helicon neurons connect to the EPG cells side by side, but have opposite effects. R5 signals quieten the EPG cells, while helicon signals excite them. So at night, EPG neurons are naturally less excitable, because normally both the R5 and the helicon cells are firing together. In the experiments, stimulating R5 during the day mimicked that nighttime passive state, while blocking R5 at night made the EPG cells more responsive to visual cues. The bigger idea here is that during sleep, slow wave activity at 1 Hz rhythm acts as a timing signal for this neural filter. At night, signals from both the body's internal clock and the brain's need for sleep keep the DFSB, the R5, and the helicon neurons oscillating together. That synchronized rhythm dampens EPG responses and lets the fly focus on sleep and all that internal housekeeping. But here's the kicker. Each cycle has an up phase, where the neurons are briefly more active, and a down phase when they are quiet. These alternating states create short windows when a strong outside signal, let's say a bright light or a sudden vibration, can slip through and wake the fly. This is why a sleeping fly can stay undisturbed with mild stimuli yet still wake up quickly enough in a real emergency. I mean, we do this. Actually, I'm going to say that some of us do this. I've met people that can sleep through a rager. But it is so impressive that something so small and simple can do the same thing. In the morning, the DFSB, the R5 and the Helicon circuits are no longer locked in rhythm. They operate more independently so the fly can fully respond to sights and sounds again. But at night, the DFSB provides that extra inhibition that helps keep the R5 and Helicon in sync, while other neurons linked to the circadian clock reinforce the slow wave pattern. And this system is flexible too. A strong external cue can disrupt the synchronized waves and reopen the sensory gate, waking the fly. But even the EPG neurons can send signals back to get the fly to fall back to sleep. Slow wave activity works kind of like a nighttime timer that coordinates different brain regions so the fly can sleep deeply yet remain ready to wake if something important happens. I'm going to be honest with you, I definitely had to read this paper and other papers just to understand what was going on. But understanding it does make me appreciate fruit flies a little bit more, even if they do ruin my appetite a little. A special thank you to all the scientists, grad students, undergrads, technicians, and any other professionals that nearly went cross-eyed looking at tiny neurons and helped create this incredible piece of science. And thank you for listening and following this very dense but exciting paper. If you're celebrating your birthday today, I hope your brain is having the best day. And if you're not feeling all that good today, I wish you an abundance of feel-good neurotransmitters. But if you're just out there diagnosing yourself with a sleep disorder based on LED lights, please like and subscribe. I'll see you soon. Peace.